Assalamu alaikum. Welcome to this session. Today I am going to discuss about the enzyme. Catalysts are the substances that increase the rate of reaction without being changed in the overall reaction process. Whenever this catalysis occurs in the biological system, this substance is called enzyme. So, enzyme is the catalyst of biological system, that is the living system. What will be the other properties of enzymes? Enzymes are biocatalyst that is synthesized in the living cells. They are protein in nature, except the ribozyme, that are the RNA molecules. As they are protein in nature, so they are colloidal, the larger particles, and non-dialyzable, thermolabile in character. That means in extreme heat, the structure of the enzyme become denatured, and they have specific isoelectric pH, and they are reaction specific. They are specific in their action and remain unchanged after the end of the reaction. The overall function of the enzyme is the acceleration of the rate of reaction. It accelerates or enhances the velocity of reaction many times than the reaction that occurs in absence of enzyme. As for example, the hydrolysis of protein outside the body at 100 degree centigrade temperature requires several days for its hydrolysis. But in the living system, that is in the gastrointestinal tract, it occurs within several hours. It is only due to the presence of enzyme, and without enzyme the life is just impossible. Let us know about the specific sites of enzyme. It is an enzyme, and it is the active site. Active site is a particular site of an enzyme. It is a cleft or pocket like site of an enzyme. It is a three-dimensional shape where the substrates can bind. It is the substrate. And the enzyme can catalyze this substrate, and ultimately it forms enzyme substrate complex. This enzyme substrate complex leads to the formation of product. It is the ultimate goal of enzyme catalyst reaction, is the product formation. It is the holoenzyme. Holoenzyme is the active form of enzyme or functional unit of enzyme. It consists of apoenzyme and coenzyme. Apoenzyme is the protein part of an enzyme, and coenzyme is the non-protein part, and usually it derives from water soluble vitamins. Whenever the coenzyme binds tightly with the apoenzyme, it is known as prosthetic group. Enzyme requires some activators, and it is known as cofactor. Usually they are the trace elements that enhance the enzyme catalyzed reaction. Enzymes are specific in their action. There are three types of specificity. These are the substrate specificity, stereospecificity, and reaction specificity. Stereoisomers are the substances that have the same molecular formula but different spatial configuration, and if each enzyme can catalyze a particular isomer. As for example, amylase can catalyze alpha glycosidic bond, and the cellulase can catalyze beta glycosidic bond. The next one is the substrate specificity. It is of two types, absolute and relative. Absolute and relative. In absolute specificity, a particular enzyme catalyzes a particular substrate. As for example, the enzyme lactase acts upon lactose. The enzyme glucokinase acts upon glucose. In relative specificity, the enzyme can catalyze a particular group or particular bond. As for example, the enzyme trypsin can catalyze peptide linkage. The last one is the reaction specificity. A substrate can take part in different types of reaction. As for example, amino acid. An amino acid can undergo transamination, deamination, racemization and decarboxylation. But the enzyme of each reaction is separate, is different for catalysis of each reaction. The unit of enzyme activity is international unit and CATL. CATL is a recent unit of enzyme activity. According to this international unit, it denotes the conversion of 1 micromole of substrate per minute. According to this international unit, it denotes conversion of 1 micromole of substrate per minute. And according to CATL, it denotes conversion of 1 mole of substrate per second. Let us know about the nomenclature and classification of enzyme. Previously, there was no particular rule for naming the enzyme, and it was named by the discoverers according to the discoverers of the enzyme. And some enzymes like pepsin, trypsin, chymotrypsin, this group of enzymes do not convey any information about the substrate upon which they act or the reaction type that they catalyze, and these enzymes are known as trivial enzymes. To remove this ambiguity, in 1964 the International Union of Biochemistry and Molecular Biology proposed a systematic classification of enzyme. According to this classification, enzymes are categorized into 6 classes. These are oxidoreductases, transferases, hydrolases, liases, isomerases and ligases. But in general, the suffix ase is attached to the substrate for naming the enzyme. It is known as recommended name. So, enzymes are classified in general into 2. The number 1 is the recommended name. Here the suffix ase is attached to the substrate for naming the enzyme. Suppose lactase, lipase, urease. Another more acceptable, the systematic name. is the IUBMB classification. According to this classification, enzymes are classified into 6 major classes. The first class is the oxidoreductases. It catalyzes the oxidation reduction reaction. These are the two substrates, substrate A and substrate B. It's the reduced form, it is in reduced form. And oxidation reduction reaction is a coupled reaction. Whenever a biomolecule becomes reduced, it just donates the hydrogen to the next one to become oxidized, like this. The enzyme cytochrome oxidase and the dehydrogenases fall in this category. The next group is the transferases. This group of enzyme just transfers groups. Suppose it is the substrate A and substrate B. Here only the group transfer occurs, like this. Transaminases are the two important liver enzymes, are the example of this transferase enzyme, this category. These are the alanine aminotransferase and aspartate aminotransferase. The third group is the hydrolysis. Here hydrolysis occurs to split the bond. The water molecules can hydrolyze this biomolecule, like this. The lipase, urease, alkaline phosphatase fall in this category. The fourth group is the lyases. This group catalyzes the breakdown of carbon-carbon, carbon-sulfur and carbon-nitrogen bonds. The reaction proceeds like this. Aldolase, carboxylase are the examples of this category. The fifth group is the isomerases. It causes isomerization of the substrates. Triose phosphate isomerase is the example of this category. The last one is the ligases. It forms a ligand. Here forms a ligand or condensation, and with the expenditure of ATP. These are two substrates, substrate A and B, that form a ligand with the expenditure of energy. So its name is synthetase. DNA ligase, glutamine synthetase are the two examples of this category. So that's all about the properties and classification of enzyme. Thanks for watching. Assalamu alaikum.